Hi, my name's Guy Riddyhoff. I'm one of the editors at Science Magazine, and I'm here today with Titia de Lange from Rockefeller University in New York. And we're going to talk about the problems that eukaryotic chromosomes have at their ends. And Titia's going to tell us why that's so.
Well, so the eukaryotes all have linear chromosomes, and it's not clear why that happened or how it happened. But there it is. We have linear chromosomes.
And because all prokaryotes, or most prokaryotes, have circular chromosomes.
And they do fine with that.
Right.
It's better for DNA replication and so on. And it's also better because of the following problem. Prokaryotes, but also eukaryotes, have a system to look for DNA damage. And this system in our cells constantly monitors the DNA for DNA breaks. Those would be very bad, of course, and need to be repaired. But in addition, when our cells experience DNA breaks, the cell responds in a way that stops the cell cycle so that presumably repair can take place. So if you think about this system, it's really quite vigilant. It's thought that it can find one DNA break in our genome, and at the same time, there are all these chromosome ends. Now, if the system would recognize the natural chromosome ends as sites of DNA damage, then all hell would break loose, because it would signal, cells would arrest in their cell cycle, and the cell would try to repair these ends. And what would happen is that it would ligate one chromosome end to another. This is bad stuff. So, at the time that linear chromosomes evolved, this DNA damage response system was already there. And so, at the same time that linear chromosomes resolved, the cells must have evolved a solution to this problem.
And what is the solution? There are a couple of answers to that question, I guess. What's the first answer?
Well, the first answer really was identified by Barbara McClintock and Miller, who realized that the end of a chromosome is different from other parts of the chromosome, and that without what they then named the telomere, for telos, Greek for end, mere, everything in a chromosome is called a mere, the telomere differentiated for the cells that this was a natural chromosome and don't mess with it. And so then the question became, what is the telomere? And Liz Blackburn, when she was with Fred Sanger and others in the late 70s, early 80s, found that it has a specific DNA sequence. A very boring sequence. Telomeres are probably the most boring part of the genome because, for instance, our telomeres are made up of about 10,000 base pairs that go TTA, GGG, TTA, GGG, drones and drones. It's like a Gregorian chant of the genome. Very boring sequence. And yet, this sequence is it. This is the sequence that allows cells to say, this is a natural chromosome end. No panic here.
So the end still has to be disguised, I suppose, is the only way you could put it, from the machinery that you described that's present in all prokaryotes and all eukaryotes. And in that transition from going from circular to linear chromosomes, there has to be a mechanism to basically secrete it away somewhere. And so what is the nature of that machinery or system?
So what happened, I think, during evolution is that first chromosome ends were a little sloppy. At some point, telomerase evolved, and that's the enzyme that makes that repeat sequence. So telomerase is a very important enzyme because it solves another problem associated with chromosome ends, and that problem is that you can't fully replicate the end of a linear DNA. the replication machinery evolved in the context of a circular genome. And so it never, experienced the pressure to deal with an end. And there is a problem. You can't finish the job at the end. And telomerase is one of the ways to make up for this problem.
By not being able to finish, so the consequence of that would be that if you didn't have telomeres, the chromosomes would in effect get shorter and shorter and shorter after each round of replication.
Yes, indeed. And that's what you see when you knock out telomerase, when you take it away in yeast. First was done by Vicky Lundblad, Jack Zostek, or in mouse cells, as done Carol Greider, Rhonda Pino. You see that the chromosome gets shorter and shorter at the end. Very gradual, progressive shortening. So you need telomerase to counteract that, and we do that in our germline. We keep our telomeres intact. And the good thing about telomerase, there are many solutions in principle to this end replication problem. Every virus with a linear genome has this problem, and they have different solutions. Adenovirus has a protein hanging on to the end that can prime DNA synthesis for the other side. There are many solutions. The great thing about telomerase is that it dictates the sequence at every chromosome end. So once you evolve telomerase as a solution for the end replication problem, now all telomeres have the same sequence. They're all TTAGG. They're all equally boring. They're homogenized. And so at that point in evolution, it became possible to evolve proteins that recognize the sequence, and those proteins would bind to all telomeres. And what happened is that these proteins evolved, and they became essential for the protection. So in our telomeres, there is a protein complex that we call shelterin, just 6 proteins, very specific for this sequence, and they do this amazing job of hiding the chromosome end, those TTAG repeat ends, from the DNA damage response.
So, just to illustrate how dangerous a naked end is, if you remove this complex, this sheltering complex, what are all of the different things that the end of the chromosome could be exposed to in terms of inappropriate processing?
It's a nightmare. When we started working on this problem, I thought 1 or 2 things might go wrong. And recently we took the whole complex of telomeres and we found out that there's 6 or 7 pathways that get activated. So here we go. You get DNA damage signaling. Alarm goes off in the cell. The ATM kinase and the ATR kinase, the 2 main transducers of DNA damage, get activated, cell cycle arrest. That's one. And then you get the repair machinery that starts acting on these ends. One thing is that non-homologous end-joining occurs. This is a pathway, it's a simple ligation pathway. It is the 2 ends, take the 2 ends, glue them back together. So what you see in the cells without sheltering is that all these chromosomes, they all get glued together. That's one thing that goes wrong. There are 2 forms of this pathway, classic and alternative non-homologous end-joining, both get activated. Then there is homologous recombination. So telomeres can start invading each other. That starts happening. And then there's resection. So there are nucleases that are important to repair DNA breaks. Eat it in, make a nice little start for homologous recombination. They start eating at the telomere and they eat the thing away. So if you think about it, sheltering is it's 6 proteins, 7 in the mouse, and there are all these pathways. It protects again. And it isn't just that it's one solution. We used to think about telomeres, sort of like a molecular condom. I mean, sheltrin would form a cap over the telomere.
Yeah.
It's really protected. that's not at all what's going on. Children is compartmentalized, and different proteins take care of different pathways. So it's much more like Charlie's Angels. You know, they all have their own gun pointing in a different direction. And the big question now is, we sort of know who does what. My 6 angels in children, they have their different tasks. The question now is, how? How do you do this? How do you block a kinase from seeing the telomere for what it really is, a DNA end? TTA repeats or not, right? It's just a DNA end. And the ATM kinase gets activated by a protein complex. It just recognizes DNA ends, regardless of the sequence. So how does it do that to protect this DNA end from being recognized? That is the big question we're struggling with right now.
So, as well as the complex, the telomere end also has a specific structure.
Yeah, what we found is that one major clue for one of the pathways, one of the two of the pathways, we think we know how it works. These are 2 pathways that both look for DNA ends in the genome. The ATM kinase looks for DNA ends, and non-homologous end joining, this ligation pathway, also looks for DNA ends. So as soon as they're a DNA end, no matter what the sequence, boom, they'll get active and do their thing. We know that in children there's one protein in the whole complex that is dedicated to these 2 pathways. And we now have an idea how it actually does this, how it represses the 2 pathways. And it came from an observation that we made together with Jack Griffith, who's an EM microscopist at UNC. And Jack and I were collaborating, and at some point he said, you know, tell me about telomeres. Didn't really know that much about telomeres. Nobody knew much about telomeres. It was not really on the horizon in those days. He said, tell me what they are, and I drew a telomere on my whiteboard, you know, nice linear thing, and he said, how do you know it's linear? And I said, well, I know it's linear because that's the way I draw it. And so Jack and I realized that we had no idea what the actual structure was of the telomere, and we decided to go and look. And this was an impossible experiment. I mean, it was really, this is one of the craziest experiments Jack and I ever have done. But we managed to look, Jack managed to look at the structure of the telomere, and it wasn't linear. He found these lariats. It was a big loop, gigantic loops. So the end wasn't out, it was stuck in. So we thought, well, maybe this is really a solution to the end protection problem. Because if you hide the end inside the telomere, then the ATM kinase that looks for ends and non-homologous end joining, they would have no idea there is an end there. So we also found that TRF2 by itself in vitro could do this loopy thing. Not very well, but it could do it. So the idea was that maybe this is how TRF2 works. Now TRF2 sits on the telomere in children and in some way has the ability to take the end and tuck it in. Now the key experiment to test that model is, of course, to delete TRF2 from cells and then look whether the loops are gone. Right? So we try it. For about 14 years we tried. And it was an impossible experiment to do by EM. Poor Jack, he did EM after EM with us because EM is not a very quantitative type of analysis. And to do the EM, we needed to purify the telomeric DNA away from the rest of the genome, because if you look in EM, you can't tell what the sequence is you're looking at, right? It could be any piece of DNA. And to purify the telomeric DNA, we would run a biogel column that was meter long. This is not a quantitative technique. And if you do a, deletion experiment, you want to be able to count molecules. So we were stuck. We just could not get to test our model. In the meantime, the model became the dogma in the field, and everybody wrote in their introductions, TRF2 makes T loops and this protects telomeres. And I became a little desperate because I thought if we ever show this, we won't be able to publish it because everybody will say, well, we already knew this. Anyway, finally, there was a solution, and this was storm imaging. High-resolution imaging developed by Xiaowei Zhang at Harvard. And I contacted Xiaowei. She was enthusiastic about the idea to look at telomeres directly without purifying them in cells. So we went to look, and it was a disaster. We looked at telomeres in cells, and the telomeres were these tiny little nubbins. You couldn't see any structure, and it's a beautiful technique. It's very high resolution. So depression all around. And then there was a postdoc in my lab, Uli Doksani, who's just fabulous, who spent 2 years developing a technique by which he could relax the telomeres out. So he was able to let them relax out, and then we could see the loops in cells. And then we used that technique on cells with TRF2 and without TRF2. And lo and behold, if you take away TRF2, the loops open up. So now, after 15 years, he finally came to the point where we can put some meat on this model.
You must have been relieved.
I wasn't relieved until it was published.
Of course. So another thing that's interesting about telomeres, and perhaps a little more controversial, is their role in aging. Do you want to say anything about that?
So it's very clear that telomeres shorten with cell divisions, and we go through a lot of cell divisions, different in different organs, as we age, and the telomeres get shorter. It's been established in the early 90s. I think Carol Greider here at Cold Spring Harbor actually was the first to provide data on that, and so this happens. The question is, what does it matter, right? Do we age because of our telomeres? Are my wrinkles due to, shortening telomeres here? And, what you really need is a genetic experiment, right, to figure this out. And so what Carol did and Ronald Pino did is they knocked out telomerase in the mouse, and then the mouse telomeres shorten just as much as human telomeres do. Now, mice start out with very long telomeres, so you have to wait for a long time and breed them over the generations. But eventually, they show aging phenotypes. So yes, telomere shortening can induce aging phenotypes, and lots of things go wrong in the mouse. They become sterile, they have liver problems, on and on. But that's the mouse. Okay? So what happens in humans? Is the mouse model relevant to humans? And many mouse models are, not against mouse modeling, but the question remains. And then in about 15 years ago, there was a breakthrough in this area because Kathy Collins was the first to show that there is a disease, human disease called Keratosis Congenita, that is caused by an insufficiency in telomerase. So these people have defects either in the telomerase complex itself, in the RNA component, the reverse transcriptase, or in the biogenesis factors, and they end up with a lowered level of telomerase. It's not absent, it's low. Over the generations, you see the telomeres getting shorter in these families, and the onset of the disease is earlier and earlier, clearly caused by telomere shortening. So the question is, what do these people have? What are the symptoms? The main symptom is bone marrow failure. So in the bone marrow, telomeres really matter because a little bit less telomerase, shorter telomeres your bone marrow peters out. These people are then treated with bone marrow transplants. It's a very dramatic disease, and then they survive longer, and they get some other things, liver cirrhosis, lung fibrosis. You know, there are some very serious disease phenotypes. But wrinkles, not so much. It's not progeria. These are not old people at the age of 4. So it's not a general aging phenotype. And it sort of tells you what you can expect as a result of your telomere shortening. So your telomere shortening could affect bone marrow as you age, lungs, liver, all fairly reasonable to assume that. But the brain, not so much. These people don't get Parkinson's, they don't get Alzheimer's, they don't get cardiovascular disease, they don't get wrinkles. So my view is that telomeres do probably determine aspects of aging, but it's organ dependent.
Right. And then maybe be interesting to finish up with cancer and telomeres. There's a link there too, as well, isn't there?
Yeah, there's an important link. We've known about this forever. So telomere shortening occurs in normal human cells, and at some point, the telomeres get so short that the protection doesn't work anymore. The cells now think they have broken ends, and they stop dividing. This functions as a great tumor suppressor system. It works well in our system, and recent genetic data argues really in favor of that. We are protected from cancer to a great degree by our telomere shortening. But like any tumor suppressor pathway, it fails, and cancer cells find a way around this, and a way they do that is by reactivating telomerase, the enzyme that maintains telomeric DNA. We've known this since 1994, 20 years ago this was discovered. And it's across the board. You see telomerase in all cancers, basically. Some exceptions. Most cancers have telomerase activity. For the longest time, we did not know whether this was actually a selected event during tumorigenesis or just sort of a side effect of the type of cell that is the cancer cell. Cancer cells have certain stem cell features. Stem cells have telomerase. So maybe the telomerase in cancer is just a consequence of that stemness aspect of the cancer. Recently, it was found that what actually happens, what activates telomerase in cancer, are mutations in the promoter for the telomerase reverse transcriptase. Beautiful work. Levi Garraway, Kumar's group, found point mutations in the promoter that create transcription factor binding sites. Telomerase now becomes active, whereas normally it's silenced, and this is how the enzyme becomes upregulated. And this shows you that this is a highly selected for event, and that confirms that telomere shortening is really something that most cancers worry about, because they select for something that acts against this pathway. And it's becoming clear that in many tumors, this is a very, very frequent event, these promoter mutations. There are probably other mechanisms by which telomerase is activated, and a lot of work is being done on this right now. So, yeah, telomere shortening is a great tumor suppressor pathway. You and I are sitting here because it works, but, you know, cancer is clever, and it gets around.
Titia, great. Thank you very much.
Thank you.
Really enjoyed this chat.